Hello, everybody. Welcome aboard. We are going to look at algae beads this afternoon and also investigate how they can be used for photosynthesis and respiration. I'm really glad to see everybody here. My name is Karen Davis. I'm working right now with Bio-Rad a little bit as a Bio-Rad fellow. I was a teacher for many years. I did AP biology, forensic science. I did a little chemistry, a little bit of biology, AP biology, as I mentioned. And this photosynthesis with algae beads and the respiration with algae beads, both is something near and dear to my heart. And I'll explain a little bit later why that is. Lee Brown is here with me as well. She's from Bio-Rad and she's kind of running as the host, and you'll see her in there and you'll see her camera view sometimes periodically. So you've got your camera view as well, Lee?
I do. So you might have to pin it in order for them to see it. So I'll try to demo things on that. Can you see my hands?
Yep.
Awesome. So, or I can pin it. You don't need to do that.
That's right.
There we go.
Okay. So we're going to switch this workshop agenda around just a little bit in order to make sure that you guys can have an opportunity to do some data collection with the algae beads that you were sent. I know that Lee had sent you some algae beads. I'm hoping that ahead of time you did the rinse and you activated the algae beads already. So I'll talk about that and the student lab materials. So with this lab, what you're given is a little brown jar with algae beads. You're also given a container of indicator, and the indicator has to be diluted. It says to dilute it like 3 days before you do your lab and leave it set out in the atmosphere so that it can acclimate. So this is mine that's been sitting out for a little while. It's kind of this amber color once you've got it sitting out.
I was going to interject a good tip here that I just heard. So Damon, what he said, if you don't have 3 days, because we're always like doing things the day before, just take a couple of beakers and pour them back and forth, and that will kind of aerate the indicator a good bit. So that works like same day, easily, like within a few minutes. So
good to know.
Yeah.
So I've had that before where I didn't have time to do it, especially if I get to a summer institute and the algae beads just got there ahead of me. So I have to hurry in and do this the first day so that they're ready on day 2. So what you're given in the kit, in addition to the algae beads and the indicator, is you're also given the little cuvettes that you use for housing your algae beads during the experimentation. You're given some disposable pipettes, and then a very important piece is this indicator card. The indicator itself is a pH indicator, and so we'll talk about why the pH changes, of course, when photosynthesis and cell respiration are going on. So you'll need this little pH indicator card as well. There's a bunch of ways you can do extensions with this lab, and we'll also talk about what those extensions might look like for you. All right, so we want to be able to demonstrate both photosynthesis and cellular respiration using a single organism. I'll delve into why that's so important and near and dear to my heart in a little while. So we're going to use our indicator solution to show how photosynthesis and cell respiration occur in the same organism. So I want you to think for a moment about the equations for cell respiration and for photosynthesis. There is a product in cell respiration, and there's a reactant for photosynthesis that, when present or absent, can change pH. If you think about what that might be, there's a hint on the page. It's carbon dioxide, right? Because carbon dioxide in solution will tend to form carbonic acid, and carbonic acid will then lower your pH. And as you see from this indicator, the gold area of the indicator really shows that area where it's changing the pH to a lower pH. As organisms are doing cell respiration and producing more and more CO2, the solution becomes more and more acidic. On the other hand, if the organism is doing photosynthesis, they're removing CO2 from that solution. And as they're removing the CO2 from the solution becomes more basic because they're removing that carbonic acid. And just a note here, your students might really struggle with the idea of acids and bases. I've seen indications on the exam that they really have a hard time understanding that the higher hydrogen ion concentration, the lower the pH. They also have a tendency to call those proteins and not protons when they're discussing anything. Same thing whenever they're talking about electron transport chain, they have a tendency to say proteins when they mean protons, or vice versa. If you have a spec 20, you can also use a spec 20, set the absorbance at 550 nanometers, and you could use that to collect some quantitative data instead of just using this indicator card. Okay, so we're going to go ahead and get started with your portion. I mentioned here again in the dark, the plant is likely to be doing cell respiration because there's not enough light. And in the light, it's more likely doing photosynthesis. So adding CO2 down here at this side, removing the CO2 on this side. And so that's a really good point to make for your students. The initial indicator solution starts out somewhere neutral, in that area of somewhat neutral. It's not really neutral, it's a little bit more basic. So it's this amber colored, which puts it somewhere in the range of about 8.1 is where mine normally starts. What you had to do before you did your activation of your beads, or before when you got your beads, you were asked to first to cut little transfer pipette. That makes it easy to catch the beads. You can scoop the beads if you cut it at an angle, or if you just cut it off square and you use the pipette tip to kind of attach to each bead, you can suck each bead and move each bead one at a time. You can only move them one at a time that way though. And if you cut the transfer pipette at an angle, you're able to move several of them. So what you're going to want to do is to transfer the beads into cuvettes. You're also instructed to rinse the beads first. So when you first get them out of your kit, what you would do is put them in distilled water for 5 minutes. You can add the distilled water right to the cuvettes. You can also pour the algae beads into your cup of distilled water, and after 5 minutes, then transfer them to the cuvettes. Just take the water away. So I'm hoping that everyone has washed their beads yesterday. Everybody washed their beads yesterday. And your beads, hopefully overnight, you added some indicator. So put 10 beads per cuvette. I think your instructions yesterday probably told you that. And then you add some of that indicator that's been sitting and acclimating, or that you acclimated. You add 1 milliliter of that indicator to each of the cuvettes. Now we're ready to begin our experimentation. So we're going to put those under the light. Okay, so I'm going to ask each of you, if your algae beads had been activated, you do want to put fresh indicator on. So if you haven't done so already, you'll want to switch out the indicator that they were activated in. Just draw that out, put it in another container, and then add a 1 ml of the fresh indicator. You'll do that to your 2 cuvettes. So I'm going to give you a moment to do that. Remove the old indicator, add fresh indicator if you haven't done so already. Lee, was that part of their directions before we started today?
Yes, it was. Oh, sorry. It was, but go ahead if you haven't done that already. You're just gonna change out the indicator, and mainly that's because you know, the goal here is to see that color change in one about 30-minute class period, right? Hopefully you have more than 30 minutes per period, but 30-minute lab period, let's say. And so putting them in fresh indicator will help that. We want to kind of start them off in the middle of the pH scale here. So you can also give them to them in water if you want. If you want to, after you've activated the beads and then taken them away from the light, you can switch out the indicator for them and give the students the beads in water, and then they can draw out the water and add fresh indicator. Both of those options work.
Great. All right, once you have created your cuvettes, you'll have 2 cuvettes, one that we're going to leave in the light, and the other we're going to wrap in aluminum foil so that you have it in the dark. So the initial lab experiment with this particular one, before the kids do the inquiry, is light versus dark, so that we can compare what happens when an organism is doing either photosynthesis or cell respiration. You'll notice that the cuvettes are laid on their side. both of them under the light. One happens to be wrapped in foil. The other one is not wrapped in foil. And Lee is showing that right now. Is everybody pinned to Lee, her camera, so you can see it here? I'll pin you. You can also make Lee, her camera view larger. So what's going to happen now is we're going to leave these under the light and dark, and then students would take a time course. Every 5 minutes they would record. So our initial, I forgot to ask you to record what your initial color was. If yours was like mine, it's about 8.1, right? So look at your indicator. Yours is also, Lee, about 8.1?
Mine looks about 8.1, yep.
Okay, so you want to record that initial. And then I'm going to set a time course so we can record every 5.
That's good. I also set up a PCR tube, and I think Karen will tell you about that later. So you can do that now or whenever.
Yeah, so that's another one that you can do as well, is setting them up in a PCR tube. I'll show you the PCR tubes, and if you guys want to experiment with it and play with it, you can. Once you have them set up, you'll want to put them both under a light. Again, 15 to 25 centimeters away. An LED light works well. A cell phone camera works well. I have mine under my vanity lights in the bathroom, but you have to make sure the lights don't produce a lot of heat. Heat will, of course, affect your results. So if you have a light that's like an incandescent bulb and it produces a lot of light, you'll want to put like a heat sink in between. So maybe a beaker full of water in front, and then the cuvettes behind. You'll also notice the cuvettes are laying on their sides rather than standing up. That's what, so that the beads can spread out and the solution can be dispersed as well.
I found this super cool book light for like, what was it, $8 or $9 on Amazon.
Yep.
It's tiny, it's light, it recharges, so you don't have to string plugs across the classroom. And it's also got like warm and cool light, so they could do some experiments with wavelength, or I guess I'm not sure what wavelength it is. They might tell you on there, sometimes they do, but in 3 different intensities as well in there. So pretty cool. And these LED lights are nice because they don't get hot, like Karen said. So if you're really in a rush, you can put them just straight on top of the cuvettes.
Yeah, I'm going to put mine down lower so that it's even more intense and see what happens. All right, has everyone got theirs set up so that they can put it in front of the light? Again, you can use your cell phone camera or some cell phone flashlight to work as well. I've already got actually the time course started, so I'm hoping that you guys have already got them all set up. We'll take a reading in another 3 minutes or so, and you can see if they're starting to change. All right, so every 5 minutes, the students would take a reading. To take a reading, they would just compare the bead solution in the light to what the bead solution in the dark looks like. Try to figure out either the absorbance if you're using a Spec 20, which is why they're in the cuvettes, so that you can use a spectrophotometer. Or you can use the indicator card. It's pretty straightforward. You can find out what the approximate pH is by comparing the color. So we'll do those time course intervals every 5 minutes. In the class, the students would use this initial experiment to find out what happens to photosynthesis over about 30 minutes if the organism is doing photosynthesis or cell respiration. We've got about 2 minutes for our next one. So the data table looks something like this. Data with the light and the dark. So you can do the pH readings to see what happens to the beads in the light and what happens to the beads in the dark. You'll notice they have minus beads in light and dark. Those would be a negative control. I wouldn't expect the solution to change color, but we could find out. Great negative control there. All right, and then their graph would be a graph of the time and then the pH changes or absorbance, depending on if they had that. Any questions so far from the group? I'd like to get into the whys in a moment here. You should see something like that. I wasn't meaning to show you that slide yet. Okay, so a couple of reasons why the students would find this relevant. One of the things is if you start talking about algal blooms and what happens, So this is a great way to introduce this lab. By the way, I did this backwards on purpose because we only have an hour here together. I did the student prep part first so that we could start collecting our data. And that now I'm going back and doing what I would normally do initially to introduce this to the students. So you see something going on here in this slide. You might ask and pose the students, what do you see? What are you observing, right? Hopefully they would notice that there's a whole lot of dead fish on this beach. And propose the question of why there might be so much dead fish, right? And a lot of you teach about dead zones, perhaps. We live here in Texas, Lee and I do, and we have to worry about the water temperature, say in a closed body of water like a lake. The water temperature gets so hot that it doesn't hold much dissolved oxygen, and you'll actually have the fish die. Okay, here you see along the coastline an example of a dead zone off the coast of Louisiana. And this dead zone is an area where there isn't a lot of algal blooms or algae anymore. And you'll see this dead zone here coming off of the Mississippi Basin. So you can go back to the why. Why would that be? So what happens, and I know that you teach this likely in your ecology classes, so it's a nice tie-in with ecology. What happens when we have an algal bloom? Okay, so there is a lot of algae initially, but of course there's so much algae and a lot of the algae will die. And as it sinks to the bottom and decomposes, it brings up the number of decomposers. Lots of decomposers are there and they're using up the oxygen in the water. And so the low oxygen is what causes the fish to die. So the fish don't have enough oxygen to breathe. So I'm sure you've taught about things like that in your ecology section of AP Bio, or even in regular bio as well. What happens if you have an algal bloom? And like I said, here in Texas, the lakes and ponds get so hot, sometimes the water gets too warm and also the dissolved oxygen goes down. It can also kill off your algae for that matter. So you can have that conversation with your students about this area of low oxygen or hypoxia and where might that come from and why would that be important, right? To make sure that the algae is maintained at a level that the environment can handle too. So that's another thing to be thinking about. This is an interesting one. What happened in 2020? If you'll notice, we kind of had a trend going here. No data was taken here. You might be curious as to what happened in 2020 that happened. Well, what happened in 2020 was there was a hurricane that came through that area of the Gulf. And of course, once that happens, the water gets all disturbed and stirred around, and that actually brought the level of oxygen up in the water. So any kind of water that you have that's flowing, like river water, generally stays oxygenated, right? It's the water that's still, that's not moving a whole lot that you have to worry more about with hypoxia. So dead zones worldwide, you can see where there are several different dead zones where there is low oxygen. All right, a second reason why this is also really important, I think, for students. Oops, and I missed my timer. We should be taking a reading. So I'm going to have you pause. I'm going to grab mine and I'm going to take a reading as well. Let's take a reading and evaluate. Unfortunately, I have to go to the other room.
I'm going to say mine's about at 8.3 now. Let's see if I can get a better view of it from here.
My algae's been going crazy because I have quite purple. I am already at an 9.1, so my light must be pretty intense. That's my light one. We need this indicator to go way out.
And then my dark one is probably at 7.5 now.
This is my dark one. Y'all can see the dark one here. Here, I'll hold them side by side. The purple is the light, obviously, and then the gold is the dark. My dark about 8.5. Is that what yours is also, Lee?
My light one's probably 8.3. And my dark one is around 7.5, I think.
Okay. All right, everybody hopefully got your time course for number 1. Let's go ahead and start time course number 2. So we'll assume another 5 minutes here.
I was going to add some, one of the things that you might have noticed, but we didn't specifically say, is if you put something white under the cuvettes, that'll help reflect the light back into the cuvettes or the tubes. And so sometimes that helps speed things along. And if your beads are super active, like Karen has, she's got them gung ho.
Yeah, I turned them on last night, and then I turned them off like first thing this morning.
Sometimes they're just like raring to go, but yeah, so sometimes they'll be really, up and at them, and sometimes they'll be a little bit more sluggish, just depending on a lot of factors. But you must have really fresh beads. So
actually, I think these are the beads left over from the summer workshops.
oh, they're really
really
good then. So you got them in May?
Yeah. Yeah, so they're 6 months old at least. Okay, so let's move on. And this is the part that is really, really near and dear to my heart. It's one of the reasons I asked Lee to please let me work with photosynthesis and cell respiration. I don't know if any of you have ever been a reader on the AP exam, but there was a problem one year that came up about photosynthesis and cell respiration that I want to share with you during this time. So what can your students tell you about photosynthesis? After doing the cell chapter, which is unit 2 for AP Bio, and then after doing the cell respiration photosynthesis or cell energetics, they should know a lot about photosynthesis and cell respiration. Unfortunately, a lot of that's not getting transferred to the AP exam. Can they connect the two, right? And can they connect it back to cells and cell parts? So for the most part, they do understand that leaves do photosynthesis, right? They understand that oxygen goes in, carbon dioxide goes out. But do they also realize that leaves also can do cell respiration? And other parts of plants also do cell respiration. It's not just photosynthesis that plants do. So in a comparison of photosynthesis and cell respiration, we generally think that plants do photosynthesis, animals do respiration. And that's unfortunately, what the students frequently respond. So they'll see a diagram something like this, with the plant doing photosynthesis and the animals doing cell respiration. Decomposers, sometimes they'll show on the respiration side as well. It's very difficult to actually find an image of the photosynthesis and cell respiration cycles. It's really difficult to find one that has animals on one side and plants on both. Even though students understand that plants have both chloroplasts and mitochondria, they're not always making that connection, right? So that most of the time they're really good at telling us what the inputs are. They're really good at telling us what the outputs are of photosynthesis and cell respiration. They understand the two processes that way, but they're never displaying their knowledge really well that plants do both photosynthesis and cell respiration. And so this came about on the 2013 AP Biology exam. And if you're a regular bio teacher, I would really love for you to emphasize this with your students as well. This came out on the 2013 AP Biology exam, and I happen to be assigned this question as an AP reader. And I will tell you that for the most parts, the students did a really great job of being able to identify that carbon dioxide switching into organic molecules and carbon dioxide in the atmosphere were photosynthesis and cell respiration. So the first part of the question asked them to identify those two processes. What were those two processes, right? So CO2 becoming organic molecules, that was easily photosynthesis. Organic molecules becoming CO2, cell respiration. The problem became the second part of the question, which said identify an organism that can carry out both processes. And it was amazing how many students could not name an organism. And I will say most of the incorrect answers said that there were no organisms that could do both. So they're not making the connection in their head that photosynthesis is done plants and any organism that can do photosynthesis, like photosynthetic bacteria, for example. But they're not making the connection that because plants also have mitochondria, they can also do cell respiration. So it's a really, really important thing to drive home that plants can do both, right? We're coming up on our second time course. So I'm going to stop talking for a moment while we check again. And we're at 5 minutes now. Everybody want to check and see what your solutions are? This is my light. It's quite purple. I've maxed out on that one.
Mine's somewhere between 8.3 and 8.5, maybe. My PCR tube, it's kind of, I don't know if y'all will be able to see it or not. Let me see if I can get it farther up there. But the solution that's closest to the beads is more purple, and the solution that is up at the top is a little bit more red. It's kind of hard. It might be too hard to see on the screen. So if that's the case, if your students get something like that, they might want to mix it up to get a better, more homogeneous solution there. But it is kind of cool to look at like that.
So I'm at like 9.1 for my light and 7.3 for my dark. So it is moving.
Dark ones.
Oh, look at your dark ones. Those are beautiful. They're
nice.
They're
looking good. I would say my dark is at 7.1.
Beads were not as active as mine.
It's because I lost my activated ones. I was telling Karen before the lab, I activated them and wrapped them in aluminum foil so they'd be good to go, and put those beads in a very special safe spot and promptly lost them. So
and Jenny, you asked about the PCR tube. I'll talk about that in just a bit, because I'm going to talk about other things you can do with these, the little beads. We're going to do the light and dark. And so we're going to go ahead and wrap them back up, and we'll start our thing again for another 5 minutes. So my point, going back to where we were here, it's really important that students figure out that any organism that can do photosynthesis also can do cell respiration. It's not always true the reverse, right? Organisms that do cell respiration don't automatically do photosynthesis, but all organisms can carry out cell respiration. And then some of those organisms can also do photosynthesis. Ironically, the most common correct answer that I had to give points for, a point for, was students who said Venus flytrap. And it killed me to give a point for Venus flytrap, because I know that's not why they're saying it, is because it's a plant. They're thinking because it's a plant that eats something.
Technically correct.
Technically it's correct, so I had to still give the point. So it's again important that students understand that organisms that have both of those organelles are going to be doing both processes, right? Cell respiration and photosynthesis. And of course, there are bacteria that can do both processes, maybe not quite this complexity because they don't have the organelles, but there are bacteria that also can do photosynthesis as well as respiration. And algae, as we know, can do photosynthesis and cell respiration. All right, so let's talk about the algae bead lab. We're taking our third time course. To show this, normally we would start the lab with the background information that I just went through with you, with the algal bloom. And generally we would then start with the pipette we're going to blow into our indicator solution. So you said about 1 milliliter of indicator solution, right, Lee?
Yeah, I think so. I mean, yeah, I would just, in case you want to use the other stuff for later. And then you can just use a straw. You can use a straw, or you can cut I just cut my pipette. I'll put it under here.
Or you can take your pipette that hopefully you've already used it for transferring your beads, and just cut the bulb part off like that. And this is a good way to introduce the lab, because you can ask the students to blow into the indicator solution and see what happens, right? So as they blow into the indicator solution, did you already blow into yours, Lee?
No, but mine is kind of yellow. I'm gonna make it more yellow.
See what happens if I do it. It's going to sound funny while I'm doing this. Hopefully you were able to see what happened. And hopefully your students would be able to see what happened as well with the indicator solution. So then you could ask questions. Why do you suppose that changed color? It changed from this amber color to this bright gold color. So ask them to make observations and ask them to start thinking about why would that. Some of them might say it's because of your breath. Some of them might say because you're blowing in oxygen. They're going to have some ideas of why that might change color, right? Then you can get into understanding about photosynthesis and cell respiration and what happens with blowing in that indicator and what about that indicator might be changed, right? So the students create an initial model and try to figure out about the indicator. Why does the indicator turn yellow? And then we introduce the idea that there's actually carbon dioxide in your exhaled air. And so what you're blowing in is then carbon dioxide. And as I mentioned, that's changing our indicator from this neutral-ish middle area. It's not really neutral 7, but the middle area of this chart all the way down to this gold area. And then we can have the conversation again about CO2, and CO2, as they might know, makes it acidic. It lowers the pH. And so once you lower the pH, you make that a solution more acidic, the indicator turns to that gold. So then you can have that conversation. At this point, your students are now ready to start with their algae beads, and they'll do their background. The first round, again, is the light and the dark. And these slides kind of talk about what happens to the indicator once again when you blow in the CO2 or the increasing levels of CO2. And I love the way this one's set up so that you can see that the more CO2 you have, the lower is your pH because it makes the carbonic acid. And then the higher your pH, the less CO2 you have in the solution. And then you can relate that to photosynthesis and cell respiration. Of course, go back to that formula for both of those, and students will realize that plants remove CO2 when they're doing photosynthesis, and they add CO2 when they are doing cell respiration. It's a good way, again, of telling the kids that the students that algae do both processes. And I should stop us again because we are on our 5 minutes again. So if you would take your beads and examine. My activated beads are extremely dark. Almost off the 9.1 scale. Let's see what my light dark ones are doing. Oops.
I'm pretty close to 8.7, maybe slightly under that in my cuvette.
Here's my gold one. I'm going to hold it up so you can see it against the. And mine is now at about. Oh, I'm still close to 7.3. My other one is at 9.1 though.
I think I'm still at 7.1 for my dark one.
Great. I'm putting them back under the light. Leave them again for another 5 minutes for those of you that are doing this at home. Oops. All right. So now we know that the CO2 in the solution is what's changing the indicator, right? The more the pH is lowered, the more carbonic acid it is. That means there's more CO2. So some experiments to go on after. What could we do that would affect photosynthesis, right? And this is where I'm going to talk about the extended piece. Even though we're still collecting our data for our light and our dark, we'll talk about what the students can do for inquiry, right? So think about the things that might affect photosynthesis. You could add a leaf to this, right, indicator, and we could actually see if a leaf does photosynthesis in this under light and under dark. That would be an interesting extension piece instead of just the algae beads. What would happen with a leaf in the light and a leaf in the dark? What would you expect to happen, right? What other things happen? What other things might affect photosynthesis? Students can then revise their model. They'll think about what happens with CO2. And they know that in photosynthesis, they have a higher pH and the solution is purple. They know in a lower pH that there's more CO2, and so the solution is gold. Now they can kind of think about those parameters. What else could you do to test for photosynthesis? Does anyone want to put some parameters or some variables I might test for photosynthesis in the chat? What variables might affect photosynthesis besides just light and dark? Pop something in the chat. Ah, temperature, correct. Temperature. And why temperature? If your students were throwing temperature out as an idea, I would want to ask them, why do you think temperature would affect it? And I'm hoping to goodness that they remember that molecular collisions are affected by temperature, right? To have more effective collisions between molecules and enzymes to be working, they have to collide. And so higher temperatures generally make things move faster, and provided the temperature is not too high, should make the reactions faster. So temperature is one. One of the tricky parts about temperature, though, you got to remember, is that this is kind of a funny one to think about. Students will think about putting it in the refrigerator or putting it in the freezer for a little while, which, you know, they could do. But what happens when you close the door, right? You have to remind them that if you close the door, you've lost your light. So they can do temperature easy. They can use a warm water solution, like a warm water bath, and they can put a cold water bath, as long as they're both in the water bath, so that the water isn't changing that, how the light reaches. I saw someone else said color of light, right? So the wavelength of light, we talk about that in photosynthesis, the fact that the wavelength of light affects how much energy is absorbed by chlorophyll and the accessory pigments. So color of light is a great one. I'll show you my setup for color of light in just a moment. What other ones do we have in here? We have temperature, light intensity. So light intensity is another great one to test. I have a little setup here with light intensity on mine. I'm going to kind of roll it around to see if you can see it there on the table. I don't know if you can see it very well. But what I did was I put my little light over here, and it's shining pretty intensely on this little guy in the PCR tube. And then I have distance from the light to show the light intensity. And you might be able to see that this one over here is not nearly as purple as the one that's way over here. And Lee was demonstrating that one as well. The PCR tube, Jenny asked earlier about putting them in a PCR tube. Using them in the PCR tube is really easy as well. Just put like 3 beads in a PCR tube and then a drop or so of the indicator solution. And so your beads can go a long ways that way if the students want to test multiple different things. Okay. So pH was suggested. Yes, pH should change the enzyme that's working, right, in the processes of photosynthesis and cell respiration. Those should also be affected by pH. But pH would be really hard to test for this one, since our card, or since our solution, is a pH indicator. So if we change the pH to test it, that would be very difficult then to compare it to our measurements on here, because now we've affected the pH. But pH is a great answer for students to throw out, because enzymes are so sensitive to pH. We would expect that those enzymes that are working with photosynthesis and cell respiration are going to be affected as well. Let's see. So Stephen asked, if you put them in hot and cold water baths, they'll get less light exposure. Correct. But as long as they're both in a water bath, like the cuvettes are submerged or standing in water, as long as both of the ones or all of the ones that you're testing are on the same parameters, they're all going to receive the same amount of light. So that was a great question, Stephen, because the light itself will diffuse some of the light. I mean, the water itself will diffuse some of that light, provided all your testing subjects, however, are in like a cold water, warm water, room temperature water, then the light reaching them is going to be the same. Tiffany asked a great question. What about pH or temperatures altering the integrity of the beads? And that could be possible if you got it too hot, right? Or even like this summer, I had some AP institutes that to put my algae beads in the freezer, and then they're not going to work. So yes, over a long period of time, in a short period of time, they could be that cold. For a long period of time, not so much. And we are at our a little bit more than our 5-minute again. While you guys are checking them one more time, I am going to grab my colored ones. I'll be back.
Mine are pretty much at 8.7, I think. for my light ones, and my dark ones are, I'd say, close to 6.9. Let me see if I can get it under there again. There's that one. And there's that one.
So I'm hoping your students, let me look and see my light and dark. I'm hoping your students will think of those same things that you guys did. And I think it's fabulous that someone also threw out pH, because that is also a variable, right? I'm going to show you mine again. You can see, I'm going to put it against the white background. You can see my purple one. He is
It's so dark.
Oh,
so excited.
That one's really doing photosynthesis. And that's my light one. And then my dark one. Let's look and see real quick. There's my dark one. And it's pretty gold now. So even though they were really pushed to the photosynthesis side, you can see that it's pretty dark gold at this point. So we're kind of right over here, and I'm probably at 7.1 now on mine. Hopefully you all have checked yours as well. I want to stop for a moment and share with you, because of this lab is set up so well for you to do these different experimental variables. Your students have the opportunity to throw out ideas of what they think will affect the rates of photosynthesis and cell respiration. You're right, temperature will affect it. Enzymes are sensitive to pH, so pH would affect it. If we could test for that, that would be difficult because of the indicator. Light color, light intensity, those would all affect photosynthesis. So this little organism, again, is really easy to use. Very minimal prep for this lab. It works out really well. I'm going to take a moment. Let me go ahead and reset my timer, so we're set back on 5 minutes for one more time course. I wanted to share with you what mine look like that I put, and how I did these for the colors. I know there are lights you can buy with colored filters, and if a student group decides to do that, you could have a setup with colored filters. But for my purposes and what I'm using, I use, these are notebooks, like not notebooks, the pocket folders that you buy that are like cellophane-y pocket folders. And I just cut a little piece out and made a sleeve. So my little cuvettes are inside the sleeves. So I have different colored on those little sleeves.
Did you just tape them together, Karen?
Yep, they're just taped together. Yeah.
Will you see if you can find that link in Amazon? See if Amazon has them. I mean, not right now, but later, and we can just send it out.
The link to the what's?
To the binder, whatever you call them. They're dividers, right? Like
they're just pocket folders things. Like, oh, I just had them here a second ago. Where'd they go? Another really cool thing that one of my groups came up with. I had a group that was testing windows, and so they were thinking this summer they did glass. They put their beads behind in a beaker, so they were behind glass. They put their beads behind like a piece of plexiglass. They put their beads behind screens. We had pieces of screen like this, and they had one screen, then they had two screens to make it darker, two screens, and then they did like three screens, and then they had one that was in behind like a wall, right, that it couldn't get to the light at all. That was really interesting to see that. So this lab lends itself to all sorts of different things. I'll just show you all real quick what my colors look like. So this one's a yellow sleeve, and my solution is pretty purple. It's almost at the 9.1. I would probably give it an 8.9. That's yellow. And this would be great too, because the students can make a prediction of what they think will happen. This color is really dark, whatever it is. Oh, it's really dark blue. And the really dark blue, here's the color of my cuvette, and it's about 8.5. This sleeve is a red sleeve. And in the red sleeve, I have about an 8.7 maybe. And so this is a great way to do it, because these little sleeves work really well. I'm going to save that one for last. This one's an orange sleeve. And orange is at about, oh, pretty good. Pretty close to 8.9. And then the last one I like to save for last is a green sleeve. And I can ask students, what do they think is going to happen in the green sleeve? Right? I'm hoping they're going to say, well, mostly green things reflect green, so they don't absorb much green. And when I bring it up, there it is. It's really close down here to this gold area. It's like at a 7.7. So it was going towards photosynthesis, cell respiration rather than photosynthesis inside the green sleeve. So those are simply just pocket folders that I cut and made little sleeves from.
That's a good idea.
Yeah, it works really well. Really well. Jen asked if this experiment can be repeated multiple times. The problem is the multiple times, you can use them multiple times, but you've got to give them a little break in between. So if I have more than one section of the classes, what I would do is I would have the students testing one period, and then maybe the other class would not test them till the next day. Or if I used a smaller setup with fewer beads in the container, I would have a different set of beads for the first period class, and the second period class would have a different set of beads. So you can use them multiple times. It's just they have to have a little bit of a rest. If you push them too far to photosynthesis, as you saw, mine were pretty pushed to photosynthesis, they don't come back to the cell respiration as quickly.
The PCR tubes are good for that. And there's a different version of the kit that has those. That way they can just do 3 beads instead of, instead of 20 beads per group, you're only using 6.
Right. And then you can use them over the different periods. You're welcome. So again, students could do some math with this one, which is always important that they do a little bit of statistics. So they would calculate slope of the lines once they have their lines. And then we have a bunch of extensions. I talked about all those different things you could do with the lab. You can also remove the beads, the algae. You can take the bead away and look at the algae under the microscope. You can test light intensity, light color, temperature, and then you could add an organism like a snail, an aquatic snail. The indicator won't hurt the snail for a short period of time, at least. So you could add a animal that you know is doing cell respiration and test it under the light as well as test it under the dark and see what happens. So there's lots of things you can do. And at that point, we're on our last time courses. So if anyone wants to check their beads one more time and see how they're doing.
How are y'all doing? Hold them up. I want to see. I always like seeing.
One's maxed itself out, so I can't keep checking on him.
Hey, those are looking good. Nice. I love it.
See them.
Yeah, I see some purple and yellow. That's great.
Wonderful.
Yeah. So the rest period is really important. You don't want to go straight from activating to
yes.
Class. You want to have about 3 to 5 hour downtime. And so that can be tricky, like if you've got first period. So you can put your things on just a Christmas tree timer, or they even now have these just like Bluetooth timers, right? Like you can run from home. Or I think Karen does this too. She just takes them home with her. So
I take them home with me. And actually, when I'm doing a summer institute, I'll take them to the hotel with me. Yeah. Put them into the light there at the hotel. Yeah, just have to make sure again that the bulb isn't too hot, because the heat will bother the little algae beads.
This little guy, I'll put under here, this one, or something like this. These work really well for activating the beads too. You don't need to have like a full-on desk lamp to activate these, just a little book light. I mean, your phone would work too, but then you have your phone stuck over it for a couple hours.
That's the little book light she's referring to. He had me buy one as well. It's just a little thin light, but it's nice because it theoretically it would clip onto a book. Right.
Susan, about the prepping, do you need the overnight charging? It really depends on the beads. That's what we recommend, because we manufacture beads like I don't know how, it depends on how many people are buying them, but every few months, let's say. And so sometimes they're like up and ready to go, like as soon as you pull them out, they'll turn purple immediately. And so no, you don't need the overnight charging. It might just take you 30 minutes. Sometimes they're a little bit older, or maybe you've used them a lot, they've been sitting in your fridge, although yours, Karen, are like 6 months old, or
Mine are
7
still
months old now, and they're still great. And so they may need a little bit more. So we recommend overnight. It's typically no more than 2 to 3 hours, to be honest. Just overnight's kind of easier, but yeah, so it just depends. So once they get to purple, that is, activated, right? And so once they get to purple, however long that takes, if it's, 15 minutes or 30 minutes, or if it's overnight, they're activated, and then you can take them and let them chill out again 3 to 5 hours. And that's only because we want to see both cellular respiration and photosynthesis. That should give you a good spread. Yeah, so we see mine worked after a 2-hour activation. Great. And these, like I said, I don't know if you heard me earlier, I lost the ones that I activated. And so I started activating these probably at 4:45, or I guess 45 minutes before we started the workshop today. So not long at all.
And these, ironically enough, I've used these multiple times. I use them all summer long. And last week when I knew I was doing this webinar, I pulled them out of the fridge and I store them I just keep them in the cuvettes, and I put them leave them in the indicator, and I just put them in the refrigerator. I pulled them out last week. I put them under the light to see if they would come back on. They came right back on. I guess I did put fresh indicator, but I put them back, and then they activated again. And then when I checked to make sure they were ready, I put them back in the refrigerator again, and they sat in their indicator. And then I pulled them out yesterday, gave them fresh indicator, and then started again. So they're amazingly resilient. I have had some dry out, and I've had sometimes where the kids overheat them, and they'll turn kind of an olive green, and when they turn that olive green, you can't bring them back. If you look at them and they're kind of an olive green rather than a pretty bright green like Kelly green, they're not going to come back. So this is another interesting thing. This is a 12-strip ELISA kit. And Lee did a really great setup of the ELISA kit where she took the book light and shined it on one end, and only one little bead per, and a little tiny drop of each of the indicator. And just with the book light on one end and not on the other end, you'll see them activate. Yeah, there's a good question about the what's happening during the photosynthesis when we're activating those beads. They've been in the dark, and they've been in a jar, in a bottle, usually kept cool. And so they're not awake, is the best way I can think of, is activating them, wakes them up, gets them started, kick starts them into that cell respiration. And then once you turn off the light, and they'll start then kind of half and half. They're photosynthesizing, and they're also doing cell respiration, but that'll help bring them back toward that middle, so that when you start, they're in this middle ground area. And that's also important why you switch out your indicator that they were in overnight, switch it with new indicator to make sure that your indicator is starting somewhere in this middle area. And that way the students see them change the indicator to gold or to purple.
I've also tried to, I'm the queen of shortcuts, because if I'm just always trying to do things faster and easier, right? And so I've tried to skip that step of washing the beads before they experiment and changing out the indicator, and it just makes the whole process slower. So it helps to wash out the old indicator that was in the beads. When you when you take out the old indicator, add water, it rinses the beads out, and that diffuses into the beads. The beads are like sodium alginate, and so the water will diffuse in and the indicator will diffuse in. But yeah, so make sure you don't skip those steps, because it just takes longer in the long run.
Right. And there's an example of what the light intensity in the little ELISA tube looked like. That turned out pretty good.
And that's just a bead and 2 drops of indicator in each little 12-volt strip. You have one now, so you can do that demo.
Yep, you could do that. And I know most of you will probably be somewhere close. Either you just finished unit 3 or you're in unit 3. So this makes a great, even if you're just past unit 3, it'd still be a really good thing to do with your students, to ask them to make observations and what do they think is happening. I think that would be really interesting. It'll also really drive home the fact that there are organisms that can do photosynthesis and cell respiration, which is critical to me.
Susan has a great question, actually 2 great questions. One is, what is in the indicator? I'm on a need to know basis on this. So I don't know. I know there's at least 2 different pH indicators, and it took a lot of doing to get the, that range is pretty narrow, right? Like it's just from 6.9 to 9.1, and to get that wide of a color variation, I don't know.
Tell everybody it's parietal.
Right, exactly. Are the beads in the light performing photosynthesis and cellular respiration? Yes. So the answer is, can you ever turn off cellular respiration, right? You could, but they'd be that's a great question for your students though, is are we only seeing photosynthesis when they're in the light? Are we only seeing respiration when they're in the dark, right? And so those are 2 different processes that they can really think about and talk about. So yeah, great question there. Yes, so net photosynthetic rate. That's right.
Net rate, that's right. They're doing more than they're doing the other.
I usually try to say it as push towards that process, right? More push towards that process, right? Something like that.
All right. Notice the slide that I have up here. Bio-Rad has terrific classroom resources, not just if you purchase the kits, but there's all sorts of these resources that you can use as well. So feel free to visit biorad.com to check those classroom resources. Anything you want to say about those, Lee?
I don't think so. Yeah, there's a lot of good stuff online about how to prep them and things. I think the PowerPoint we have the prep in, it's literally just diluting indicator from 10x to 1x and activating the beads like what you guys did. So the only part of the prep that you didn't do was, you know, a classroom's worth, but also diluting the indicator. That's it, and acclimating it, or you can port back and forth between beakers. So this is like one of my favorite labs to do because the prep is literally 10 minutes. It's real fast. And you can use them from class to class. So
Susan asked how many beads are in a jar. I'm not
about 200, at least 200.
Okay. I know there's
and that's the same for both. I think if you go a little bit farther, Karen, on the PowerPoint, I think it has the other version of the kit. Yep. So one version of the kit's called for AP biology. It assumes that you might want to use a spectrophotometer and comes with the cuvettes. The other version of the kit is the exact same, except instead of cuvettes, you get PCR tubes. Otherwise, everything's identical. Okay, so all the solutions and plastic pipettes and things are identical. So again, we call that one for general biology in case you want to do this with your 9th grade class or whatever grade you teach Gen Bio in. Just because you get more setups per kit. So that'll do 24 lab stations instead of 8 in the AP Bio one. But again, you can reuse them, so they're not going to get used up. The indicator you can save and reuse as well. Don't throw that out. You can just re-equilibrate it. No, even after you've blown it, you'll see if you just leave it out, it'll go back to red.
Well, thank you all for being here.
Thank you so much.
Yes.
I'm going to stop the recording now, and then we'll take questions.